Hello, my name is Lindsay Shuck. Thank you for joining today's Family Office Network's webinar series featuring PYC Therapeutics. Before we get started with the presentation, I would like to take a minute to introduce our speakers. First, Doug Huey. Doug is PYC's Managing Director in the United States. He is a former partner at McKinsey & Company, where he helped organizations across strategy, finance, and operations. Doug has lived in Australia for the last 7 years, where he got to know PYC Therapeutics. He joined the company this year to expand their commercial presence in the United States. Dr. May Ofrelia is also joining us today. May has an extensive background in clinical drug development that spans over 2 decades in multiple therapeutic areas with a focus on rare diseases, oncology, hematology, infectious disease, and women's health. Her most recent role was president at Oncology and Rare Disease Consulting, LLC, and prior to that, she was the chief medical officer at Cambridge Life Sciences, where she led the clinical development and medical affairs group. May also spent 9.5 years at Pfizer. At the end of today's presentation, we will have time for Q&A. Please use your questions chat option on your screen to send over any questions you have at any point during the presentation.
Thank you, Lindsay. And thank you to everyone who's joined. We're really excited to be able to share the story of PYC Therapeutics with you. Maybe before we jump in, as a public company, I just need to read this safe harbor statement. So before we begin today's call, I'm just going to try to minimize my screen here. So before we begin today's call, I'd like to make the following safe harbor statement, reminding you that today's discussion will contain forward-looking statements that involve risks and uncertainties. These risks and uncertainties are outlined in our filings within the Australian Securities Exchange. As such, actual results may differ materially from what we discuss on today's call. We disclaim any obligation or intention to update these statements in the future. All right, so with that out of the way, hey, let me introduce myself. Doug Huey, as Lindsay mentioned, spent the last decade plus at McKinsey & Company, helping companies on a variety of topics on strategy and ops. Was thrilled to join PYC earlier this year, given the exciting point of time that it's in. I was down in Australia where I got to know the company, moved back to the US to open up the office here and really help with the expansion and introduce this company really onto the global stage and to investors like those on the line right now here in the US. So that's me. May, perhaps you could take a moment just to introduce yourself a bit more.
Sure. Like Lindsay said, I'm a pediatric oncologist by training, been in the pharma industry for over 20 years and spent about 9.5 years at Pfizer building clinical development for rare diseases from the ground up and manage a number of drugs from first in man all the way through commercialization. I'm really excited to be part of PYC to shepherd our incredible science that we have in Perth and develop medicines that are meaningful for patients with inherited retinal disorders and beyond over the coming years.
Thanks, May. Now, the other folks that you see on the screen are the rest of our executive team, which are based down in Perth in Western Australia, and that's where the company's operations are held. May and I are in the Boston office, and really the purpose for us being there is to introduce the company to investors, other pharmaco that we could potentially do deals with, and then obviously the FDA is here in the US, and we have a drug that we're moving towards the clinic, so we got to make sure that we're working with the regulator on that. Just a few shout outs here. So on the top left is Sue Fletcher. Sue, she leads all of our discovery efforts, and you may not, if you're not in the biotech world, you may not have heard of her, but we are really lucky to have her. She has developed 2 RNA therapeutics that are now in market, which were commercialized by Sarepta. And you may have heard about Sarepta, a really big company when it comes to genetic therapies. So Sue has invented those drugs, and she's invented our lead program and leads our discovery efforts. So Sue is definitely worth calling out. And then perhaps Rowan, who's on the right in the middle there. Rowan leads our efforts down in Australia and really was pivotal in the transformation of this company from a little tech company down in Perth now to a drug development company that's stepping out onto the world stage. So just to give you a sense of who the people are. Now, a lot of these conversations, we are talking to big life science funds, and they kind of understand the biotech industry. I don't know about the audience here, so I'm not going to assume that. So I'll try to describe a little bit about the economics of a biotech. Apologies if some of you are already across it. But if I start with who we are from a commercial standpoint, this is just a corporate snapshot. We are a publicly listed company down on the Australian Stock Exchange. The market cap numbers that you see here are in Aussie dollars. So in terms of US equivalent, we're about 250 million US in market cap. Cash on the books, we have about 25 million Australian, which is plenty to fund our lead program through its upcoming milestones. But we do want to grow. So we're looking at ways to unlock that growth. No debt on the balance sheet or anything like that. Other callouts here, bottom left-hand side, you can see that the top shareholders account for about 50% of the company. So we've had the benefit of these 4 individuals, just high net worth folks down in Australia, of supporting the company over the last several years, and they're committed to continuing to support us as we grow and expand globally. And then maybe just a comment on our share price performance. It's been a great year for us. Not surprisingly, given the successes we've had with our science, but we believe this is just the beginning of a huge growth profile in the future. And really, to hammer that home, I just wanted to show you a bit of a comparison. PYC is up here on the top, and you can see our market cap is much lower than a lot of other companies that we consider peers. So these are other companies that are working in the RNA or DNA space, and these are all preclinical companies or companies that have just filed an IND. And an IND just stands for an investigational new drug. It means that you've done enough work that you can go to the FDA, file this IND, and basically get clearance to start testing your drugs in humans. So the short of it, these are a bunch of companies that they don't have drugs in market. They're not making money yet. And that's where we are right now. But you can see we're at like a quarter of the valuation. Now, what's the biggest difference between us and these other peers? Well, that second column there, which is the listing. We're not on the NASDAQ, we're down on the ASX, and we believe that drives a significant discount, which could also be framed as a big opportunity in terms of an investment proposition. So that's just kind of framing up this investment. And if I were in your shoes, kind of looking at this company, I think this page is pretty important. But me telling you that is one thing. Let me show you some of the data and share our strategy and our science, and I think you'll get even more excited. And before we get into that, maybe perhaps the other lens that an investor would look at. And this is when I first got into the biotech world, it's kind of this formula. It's not rocket science, pretty much any industry. The value of PYC ultimately comes down to the markets that we're going after, the profitability once we get there, the probability of success of getting there, which is in biotech is probably the biggest driver. What is that probability? And then of course, what does it cost over the next few years as you're going through clinical trials? And then you times that by growth. It's nice to have one drug, but of course, it's great to have a pipeline of drugs. So I just want to hit you with the highlights. If you look at PYC and our fundamentals, the market that we're going after with our lead drug represents a $1 to $2 billion per year market. That's a blockbuster market. And that's based on the patient population that our drug will address and orphan drug pricing, which is considerable. So orphan drug pricing, we're talking in the $200,000 plus territory basically for these drugs, because there's just no other treatments available for these patients. And the disease is really hard. So we're going after a form of blindness, which obviously has a huge impact on life. So that's the market. Now the profitability here is incredible, 90% margins. What other industry do you get 90% margins? Now we're able to do that because the patient sizes we're going after are small. We're talking 4,000 to 8,000 patients. So it's not like we have a huge manufacturing supply chain that we have to put in place. We also don't have to put in a huge sales and marketing team or commercials on TVs. We can get to the ophthalmologists that are going to deliver our drug in a fairly cost-effective way. So $1 to $2 billion, 90% margin. Probability of success. If you look at the benchmarks for the type of drugs that we're going after, it's easily 45% chance of getting to market. Now you might say, wow, 45%, like that's less than half. Well, that's the world of biotech. And if you look at traditional drug development, that number is probably more like 10% for most drugs. So 45% is actually really high, and that's driven on the kind of drug that we're going after, which is a genetic medicine. And then when it comes down to the investment, about $20 million to get from where we are right now ultimately to market. And again, that is much lower than traditional drug development. And we have a few things in our favor. The first is that the patient sizes in our clinical studies are going to be pretty small. There's also really favorable regulatory pathways. So the FDA recognized that these are patients that are going blind. There's no other treatment available. So they want to get you through as fast as possible to get this impact to the patients. And finally, the other huge benefit that we have is being in Australia, there is an extremely favorable tax rebate. So 43 cents on the dollar of everything that we spend on R&D, we get a check back from the Australian government to help drive that domestic spend on R&D. So that basically extends our runway for several years till we get these drugs to patients. And then the last point on growth, in addition to our lead program, we have 6 other programs in our retinal pipeline, and we're exploring opportunities outside of the retina in the brain, in the liver. So big opportunities for growth. So I'll come back to this page at the end because I think it's pretty important when you look at these dimensions, and then I'm sure everyone on the phone here can do the math on what the NPV, if I said, hey, in 5 years, you're going to get $1 billion annuity for at least 7 years plus. You got a 50% chance of getting there, and it's only going to cost you $20 million to get there. And oh, by the way, there's huge growth. I think that math's going to come out to much higher than our current $250 million valuation. Okay, so that's the investment proposition. Let me get back to the strategy and the science. And I'm not going to get too deep on the technical stuff, but please, in the Q&A session, feel free to ask any questions if you want to dive deeper into the technicalities. So when it comes to our strategy and where we're playing, there's really 4 big themes. The first is we are part of the precision medicine revolution targeting rare diseases. So that's where we're playing. Now, what does precision medicine mean? It refers to genetic medicines. So we are able to go in and basically alter not the DNA, but the RNA, which is ultimately driving a lot of these genetic diseases. And so we can get into the cell, into the nucleus of the cell, and correct mutations in the genes to ultimately avoid any negative consequences of those mutations. So that's what we're focused on. And really, we believe we're at the beginning of a precision medicine revolution with the advances in science over the last few years. Now, there's a lot of people in that space. What makes us different, though, is our technology. And I'll come to your hurdle in genetic medicine is how do you get into the cell? So I'll come back to that, and that's what our technology allows us to do. Now, we're not just a technology company trying to figure out how to apply it. We have a beautiful lead program going after a rare form of blindness, and I mentioned the market size earlier. And then beyond that lead program, we have excellent growth opportunities both in the eye and beyond. So let me step through these elements a little bit. Let's start first with the space, precision medicine. So on the left-hand side, just want to bring to life rare genetic disease. Of course, you hear about big diseases like cancer or cardiovascular disease, but rare genetic diseases, when you add them all up, are huge when it comes to patient need. There are 5,000 of rare diseases with a genetic basis. 95% of these diseases have no treatment options. If you add all of these up, there's about 250 million or 300 million around the world affected by these diseases. Half of them are children, and 30% of them will die before the age of 5. So just a huge need when it comes to patients. Now, on the flip side, the system is set up to really encourage companies to try to address these diseases. And so the opportunity, a few of the numbers on the right. I mentioned before, you are 4 times more likely to get to market once you get to phase 1 if you're going after one of these diseases. That's compared to traditional drug development. The timelines to get through the clinical trials, 40% lower. The cost of these clinical trials is also 40% lower. And then when you do get to the market, the pricing is 7 times higher. And again, these are incentives because the system is set up to try to encourage companies to get to these rare diseases and try to help these people and ultimately advance the science. So huge need and a huge opportunity. Now, as I mentioned earlier, that's not a secret, and there's lots of companies playing in this space. And this slide is really just it's certainly not exhaustive, but to give you a sense of the landscape. And so there are kind of 2 big buckets here. One is who's trying to influence the RNA and who's trying to influence the DNA. And there's a bunch of different ways you can do that. Now, we're in this column here called antisense oligos. And antisense oligos are really just a way that you can go in there and repair the mutation in the strand of RNA. And maybe really simple, I think we all kind of know what DNA is. The DNA turns into RNA, the RNA then turns into protein, and then the protein drives all the different processes in our bodies. That's a super high level description, but hopefully you can go with it. So some of the companies here that are trying to go after the DNA, they're going in there, they're changing the DNA, and then you get that knock-on effect, and you ultimately get the right protein and get the right process. We don't mess with the DNA. That can create all sorts of unintended consequences. So what we're doing is when the DNA basically gets transcribed into the RNA, that's where we intervene. So we're able to intervene there, then you create the right protein that you need, your body performs as intended, and you don't have the disease. So antisense oligos are a way of doing that. Big companies that are doing that include Sarepta, Ionis, and Stoke, and that's the space we are in, and ultimately we want to look like those companies as well. So that's the space. Now, why do we think we can compete? Let me tell you about the technology. So I mentioned earlier, the major challenge for RNA and DNA drugs is how do you get into the cell? The cell membrane has evolved over hundreds of millions of years to keep foreign substances out. That includes drugs. So most of the drugs that all of us take, they're happening outside of the cell. If you want to treat a disease that has a genetic cause, you've got to get into the cell. You can't get trapped inside of the endosome, and you've got to make your way all the way into the nucleus where you can hit that DNA or that RNA. So that's what we can do. And just to bring it to life with numbers to prove it, here's how we do it. We combine our drugs with something called a cell-penetrating peptide. A peptide is just a sequence of amino acids. And when we basically understand what peptides are really good at getting into the cell, and we have proprietary technology around this, so we attach our CPPs, cell-penetrating peptides, to our drug, and that allows it to get into the nucleus. And the chart here shows what's happening. So this is in a mouse model. And exon skipping is what we're measuring, which basically is just saying, can you hit the gene that you want to hit? And if we inject just a antisense oligo, so this is the drug, without anything attached to it, you could see that you cannot reach these cells and you can't drive the genetic change that you need to. Now, there's different forms of these drugs. The ASO stands for antisense oligonucleotide, which I mentioned earlier. There's another form of an ASO called a PMO, which is really just a subset of ASOs with a morpholino backbone. And so same thing here, even the PMO can't get in. But when you attach that PMO to our cell-penetrating peptide, now suddenly you're getting the reaction that you want. So we can basically hit the cells that no one else can hit. We can drug the undruggable. That is our technology, simply put. Now, if I go into a bit more detail, there's those two elements of our drug. The PMO, which is on the left-hand side, as I mentioned, is a form of an antisense oligo. Now, there's other companies out there working with ASOs, but they've got a few downfalls. The first is the level of precision that they can get in terms of controlling the protein expression. The second is that those ASOs can have negative binding interactions with proteins, and that can cause toxicity, and then also stability. So the PMOs, they don't have any of those issues. So it's a really attractive drug to be working with. Now, the downside of PMOs is that they do struggle to get inside the cell, and that's where we attach our cell-penetrating peptide there in the middle of the page. And these CPPs are able to deliver the PMOs with really high efficacy and low toxicity. So it's a safe and really precise drug. So you put those two things together, you get the CPP PMO on the right-hand side, or PPMO as the industry sometimes calls it. And these are really safe and effective drugs. So that's what we're working with. Now we're focused on the eye. So maybe to give you a sense of what this looks like. On the left-hand side, the retina is a really complicated organ. There's all sorts of layers. And you can see here, if we try to put our CPP ASO into the retina, it is able to penetrate all the way through the ganglion cell layer, the internuclear layer, all the way down to the photoreceptors and the RPE, the retinal pigment epithelium. Those are the cells at the back of the eye that you've got to hit if you want to treat the diseases that we're going after. And as I showed you with our CPP, we can get there. If you go in with a traditional ASO on its own, it starts to lose its penetration as you get down to the RPE, and ultimately you can't reach the cell that you need to hit. That's the left-hand side. Now, depth of penetration is one thing, but you also need breadth of delivery. And on the right-hand side, we're showing that. So there's a couple injections that you can do to treat retinal diseases. One is intravitreal, and that's what we do. And you can see the syringe there that's basically delivering the drug to the center of the eye. And now that's the way we do it, and you are going to hit the entire retina. And it's really effective. There's another method called subretinal, and other gene therapies rely on that. Unfortunately for that, you've got to just go back to one part of the retina, and you can see AAV treatment area. That's what it is, and they call that a bleb. And that can be effective to treat those cells, but unfortunately it doesn't reach the rest of the retina. And for the diseases that we're going after, you've got to be able to hit it all. So this is a big competitive advantage for our approach. So that's the differentiation. Let's talk about the third part of our story, which is our lead program. Genetic eye diseases. So eye diseases like the one we're going after, which is called retinitis pigmentosa. Patients with diseases like this, they're born and they can see fine. But at some point in childhood or adolescence, they start to lose their night vision. And this tunnel vision effect starts to take hold. And unfortunately, over time, that tunnel just starts to get narrower and narrower until ultimately at some point in adulthood, they go completely blind. So pretty devastating. And I can only imagine what it must be like if you have a child who gets this diagnosis, let's say when they're about 12 years old or so, and they're told that they're basically going to lose their vision. So it's really hard on patients. This is just to give you a sense that from, let's say, 0 to 10, things are okay, and then it starts to onset and you start getting your vision loss. So just how it's a progressive disease. Now let's talk about what's happening at the genetic level. So a little bit of 101 when it comes to the genetics. So we all have 2 copies of each gene in our chromosomes. You get one from your mom and one from your dad. Those genes code to make proteins, as I mentioned earlier, and those proteins help our bodies function, and you can see. So that's the left-hand side, a healthy eye. If I have RP11, one of those genes is mutated, so one of the copies of the genes. And so that means I don't create as much protein as I would have if I had 2 healthy copies. So you can hear the technical term is called a haploinsufficiency. So I create about half the protein that I need, and as you can see, I start to have issues with my processes, and I start to lose my vision. What we are able to do on the right-hand side with our drug is we're able to get in there and influence one of the genes that ultimately controls for the gene that you need for vision. And we basically are able to control this gene so that we turbocharge the production of the protein. So you get it back to the levels of a healthy person. And so with the additional protein, now you can see. So we basically reverse the impact of the genetic mutation. Now, in every drug program that you have, there are risks that you've got to counteract. And I just want to use kind of this slide here, and I'll keep coming back to it, and why we have confidence that we could do everything that I just described to you in terms of correcting that genetic mutation. So the first thing in terms of building confidence in the program, and this is something we would have done last year, is did we define a commercial market that was attractive? And we certainly have, and I told you about the numbers in terms of the $1 billion per year. The second one is it attractive in terms of the clinical development timeline? And certainly it is, given that this is an orphan drug, so that means there's no treatments available on market, so it's a huge unmet need. So those are 2 big checks on that. And perhaps for time so that we get the Q&A, I won't go into a lot of detail, but this just gives you a sense of how we break down to the $1 to $2 billion in terms of how many people have retinitis pigmentosa, how many people have the type of retinitis pigmentosa that we're going after, which is type RP11. And there's about 4,000 to 8,000 people. And then if you apply the medium rare drug pricing to that, that's how you get to the number. So that's the market size. And on the favorable clinical pathway, this is just kind of bringing that to life. A lot of drugs have to go through 3 phases. We don't have to do that. We go to phase 1, and then we have a single combined pivotal phase, which is the phase right before you get approval. So a compressed timeline. So all sorts of advantages there. So those are the 2 big checks. Now, the next big hurdle that you got to get over is the scientific one, which comes to, is your drug effective and is it safe? And that's really what we've done in the first half of this year. So the first thing we did on these check marks here is we proved that our cell-penetrating peptide could deliver our drug to the target retinal cells. And we were able to do that in mouse models. The second huge milestone is that we proved that the drug worked in diseased cells. So we're able to take patients with RP11, take their cells and actually grow little retinas and take the RPE cells that I mentioned earlier, apply our drug and see if we can reverse the disease process. And we're able to do that, and that was a tremendous achievement. And then finally, you have to prove that your drug is safe, and we've been able to do that in mouse models. So that's where we currently stand. And just to bring that to life with some of the scientific data, so this chart right here, these 2 charts show that we can deliver our drug to the target cells. So this is with our lead cell-penetrating peptide. And we are delivering an ASO in the mouse, and you can see that we're hitting both the neural retina, and as we increase our dose, we see that dose response. And that is really important. You want to know that the more of this stuff you put in, that it drives a different change. And you see that both in the neural retina, which is, let's say, the first few layers of the eye, and then also the RPE in the choroid, which is the back layer of the eye. And that's ultimately what we got to be hitting. So we're really excited that we can deliver the drug effectively. The second, though, is your drug effective? And so we're back to exon skipping here. And as I mentioned earlier, this is showing, can your drug drive the change in the gene that you want? And we can. So here we've got our lead drug, the PMO or the ASO, combined with our CPP. This is the retinal pigment epithelia, so the back of the eye, those are the cells we want to target. And we can see that, again, with a nice dose response, that we can drive that exon skipping on our target gene. So great, you could drive the target gene change, but then ultimately, does that change in the gene cause a functional restoration? So let me, again, a bit of 101 on the science behind this. If I go back to the structure of the eye, those bottom two layers were the photoreceptors and the RPE. Now there's a really important process that takes place in all of our eyes. It's called phagocytosis. And it's been described to me like a snake shedding its skin. So what happens is the photoreceptors have these outer segments. And on this graph on the right-hand side, those blue bars are the outer segments. And they get handed over basically to the RPE cells that need to decompose those outer segments. So that's what's happening from stage 1 on the left to stage 6 on the right. Those outer segments get handed over to the RPE, and the RPE need to decompose or phagocytose that debris. If that doesn't happen properly, the toxins start to build up, those outer segments, and I start to lose my vision. That's what's happening in these patients. So we want to turbocharge phagocytosis. So what we did is we took the RPE cells of a patient with this disease, we put our drug in, and we said, all right, we see a change in phagocytosis. We saw the change in the gene expression, but does that result in the functional improvement? And that's this page. And again, one that we're really excited about. So on the left-hand side, those green dots, those are those outer segments that I described. And you can see that a patient with the disease, they have some level of phagocytosis. You see some green. But when you put our drug, now you see it really light up. So we're able to kickstart phagocytosis. The right-hand side is converting those images into numbers. And you can see that we're going basically from kind of sub 40 to above 60 in terms of the intensity of this process. And the beautiful news is that is enough to tip the scales in terms of avoiding the vision loss. So if we can get it to above 60, then you're basically looking like a healthy person. So really excited that we've got this level of validation in patient-derived models. So the drug is effective. Now I mentioned that third big check mark is your drug safe? And so in this chart, we are measuring the toxicity of the drug in mice. And on the left-hand side, you can see we're in the middle there, PYC, CPP, ASO, and there's just a beautiful tox profile. It's very low. We compared this to other CPPs that are out there in the market, and you can see those tox levels are much higher. So the drug is safe. And on the right-hand side, we're just measuring something called retinal thinning. If you have tox issues in an eye, your retina starts to get thinner. And so we're measuring that, and we can see on the top right, that's a good looking retina, so no thinning issues. And just to compare it, the bottom right shows one where there is retinal thinning, so you can see we have no issue. So in terms of our confidence in the program, just to recap, great market, great clinical pathway, it's effective in human models. We can prove that it's working in mouse models safely, and the CPP will deliver it to the target cells. So all check. So what's next? So now in the second half of this year and next year, we're going to be putting our drugs into additional patient-derived models. So we've tested now on a couple patient cells. We're going to expand that data set for even more patients, and we'll get a great feeling for how our drug works on different types of this mutation. We're also going to prove the safety in bigger species. So you go from rabbits, or excuse me, to mice, which we've done, now into rabbits, which is very sensitive. Then we'll go into monkeys and then ultimately into humans. So we're just now launching those rabbit and monkey studies. And then after that, or maybe just a quick comment on that. So there's a lot of words on this page, but it's basically those two big milestones that we have coming up in the next few months. So the additional patient readouts, and this will allow us to improve the design of our human clinical trials. And then, as I mentioned, the most sensitive species, which is the rabbit, which will prove that our drug is safe at really high doses, and we won't have any issues with that. So that's coming up. And then next year and beyond, we will be filing that IND. So I think I mentioned in one of the early pages that IND, investigational new drug, that basically will give us permission from the FDA to go into human trials. And then the human trials takes a few years, even at an accelerated pace. And that's where we'll go into phase 1 into humans, and then ultimately into a combined phase 2, 3, or a pivotal study until we get to market. And perhaps, May, I know I've been talking so much. Why don't you talk a little bit just about the clinical trial process, which you have a ton of experience in, and some of the things that we have going our way when it comes to clinical development.
Yeah, thanks, Doug. Well, first I want to say that I'm incredibly excited about our lead program and our ability to bring potentially disease-modifying therapy to the market for patients with no available treatment options so far. There's several advantages that enable us to expedite clinical program. The first thing is, one of the challenges of drug development is to figure out how to measure the drug's impact on patients. However, the good news is that there has been tremendous advancement in the ophthalmology clinical research space, where we can leverage what has been already done before us in the space by others with similar focus on rare and inherited retinal disorders. Secondly, we have close collaborations with the Lion's Eye Institute in Australia, and we have started building our relationships with patient advocacy groups around the United States. These organizations are quite fundamental. They have developed extensive databases for patients with inherited retinal disorders, which will really give us a head start in identifying patients with this specific mutation, PRPF31. This is really, and I'll tell you from all my experience, this is an unprecedented access to patients, is in a tremendous value for us as we get closer to starting our first in human studies. Last but not least, we are going to utilize innovative clinical trial design with seamless transition of patients from the first in human study, right into the pivotal trial, which will help us get on the market. This approach will help us bring medicines to market with little downtime between each phases of development. With that, I'll say, I think you can sense my excitement, but needless to say, I am quite committed to expediting the development cycle for our lead program, VP001, to deliver medicines for patients who need it the most. Back to you, Doug.
Thank you, May. Great. So that was our lead program, and then that's not all we've got. So we're not a one-trick pony, so to speak. So beyond the lead, this is just our pipeline page. So to give you a sense, talked about the lead, which is at the top to the human trial by the end of next year. And then on the back of that, we have 6 other opportunities where we are designing and validating drugs for other inherited retinal disease. And the beauty is that we've now built a bit of an assembly line, so to speak. We're really getting good at developing these retinal drugs. And so we'll be able to leverage all the learnings from our lead program. And that includes using that cell-penetrating peptide, which just performs beautifully in the eye. Of course, beyond the retina, we have other collaborations, and neurodegenerative disease is probably the next one. The brain, like the retina, is really complicated, and it's so important to get that depth of penetration for your drug. So we believe our cell-penetrating peptides can make a huge difference there. And then we also have a collaboration in the liver. So in some way, when we talk to investors, it's often this lead program is great on its own, but it's really going to open the door for ultimately to expand this technology and use it at scale. So those were the 4 main points for PYC. And perhaps I just come back, I introduced kind of talking about the economics of this whole thing. We then transitioned into science. But I'll just come back to this, which is really the math on the investment proposition. And hopefully we've conveyed in a clear way of the market that we're going after, how attractive that is, the profitability, our odds of getting there. And especially when you take into consideration the heavy scientific validation that we already have, the favorable pathway that May outlined, we can get there pretty quick and pretty inexpensively, and then the growth potential that we have beyond that. So with that, I will pause and happy to take any questions that the group has.
Doug and May, thank you so much. I'm going to jump back in here and just encourage everyone again to utilize your questions chat option on your screen to send over any questions that you have so we can have that open dialogue today. And to kind of get us started, Doug, we do have a couple of questions. If you can elaborate on what makes PYC an attractive investment?
Yeah, so I think the first thing that comes to mind is how we're going to create shareholder value over time. This isn't something where you need to invest and wait 5 years until we get to market. We have value creating milestones really ongoing. So in the next 6 months, if you think about the results that we're going to get in the rabbit and the monkeys, is confidence in our program. Beyond that, as we saw, that's going to further increase, start to grow, and we start bringing our new programs online and put them into these studies, like the rabbit and the monkey. Again, that's going to create shareholder value. And then ultimately, as we start to transition these things through the human clinical trials and get to cash, let's say in 2024. So I think it's really attractive from that standpoint, as far as how shareholder value will be built progressively over time.
Okay, fantastic. And kind of next question, which kind of falls in place with the first question, if you can kind of go into greater detail on the capital funding strategy.
Yeah, definitely. So we are in a fortunate position where we have enough capital to really drive our lead program into that pivotal phase that I talked about. Now, we do have the growth potential, and so we have a few options of doing that. I think as we start to validate our lead program, there's two ways. Obviously, we could raise capital and fund the growth that way. Other ways, though, are partnering with other pharmacos. And I think if you look at the headlines and you see the kind of money that pharmacos are putting into biotechs, it's just tremendous. And so if we could do some kind of partnership deal, we might have to license out a little bit of the future revenue streams on those future programs, but it would certainly be worth it to unlock the growth.
Perfect, thank you. And then what about the exit strategy for new investors? What does that look like?
Yeah, I mean, the great news is that we're already a publicly listed company, so investors can enter and exit the stock fairly easily. And I think, we're not extremely, the activity isn't huge in terms of the liquidity, but it's big enough that you could get out of the investment and not kill the stock price. So we think that creates a ton of flexibility. And then at some point, we do expect that the NASDAQ will be in our future. We'll just have to figure out the right time, and then that'll create a ton of liquidity for the stock.
Okay, great. Those were the 3 questions that we had come in so far. Again, we'll wait a few minutes to see if anyone else has any questions. Is there anything else that you guys would like to say? May, do you want to jump in at all?
Yeah, I'll say that what is exciting about this space, I don't know if folks on the line are aware of, traditional drug development for a common disease from discovery up until they get to the market is about 20 years, 15 to 20 years. Rare disease has obviously it's rare for a reason. It's very hard to study and hard to find these patients, but we have a tremendous advantage and leg up on that. And so our timing, we have 4 years to go to get to market. And in an expedited fashion, the patients have been identified. There's a tremendous knowledge in the community about the disease, which is half the battle. And we have a regulatory pathway that is well laid out for us. So I'm quite excited about this opportunity to say that, it's in 4 to 5 years. Historically, it's very hard to find the same person that worked on a drug from beginning to end. And we have an opportunity here to see the drug reach patients and to change people's lives.
Thanks so much, May. And actually, we have another question that came through. What are large biotech investors already invested here? And have these investors how have they responded to the company so far?
Yeah, so right now, half the company is in the high net worth individuals, and the other half are really smaller investors in Australia. So our goal is to institutionalize the register a bit more and bring on life science funds. So no major life science fund is currently invested in the company. But being in the US now, that's been a big part of what we've done. So virtual roadshow over the last couple months, and the response has been fantastic. And we continue to strengthen those relationships, and we expect over time to start to formalize them as companies want to come in.
Perfect. Next question. Does PYC have patent focused on the technology, i.e. transferring the knowledge of the eye to brain, liver?
Yeah, great question. I should have, yeah. Oh, sorry, Lindsay.
Oh, no, you're good. Go ahead.
Yeah, so yeah, I didn't talk about the IP estate, but it's very strong. So there's a few levels of this. So it starts really with the foundational IP of the company, which is around its library of peptides. So while most companies are basically a needle in a haystack doesn't put it right. It's like you're fishing in an ocean to find the right sequence of amino acids to give you the right cell-penetrating peptide. We've got a library of these things that have been derived by microorganisms. And so basically it's the theory that the ability to get into the cell lies in nature and in evolution. And so we're, rather than swimming in this ocean, we're starting with a pool of really effective CPPs. And so we have patents over that library and the processes that we use to screen them. And then once we have our lead CPPs defined, we have patents on those. And then we also have patents on the drug itself, so the antisense oligo. So protection on that. And then as we continue to use our library to develop other CPPs, so let's say one not for the eye, but for the brain, we'll obviously patent those sequences as well.
Great, thank you, Doug. And what about in setting up your trials? Will you target researchers and hospitals like Willis Eye and et cetera?
Yes, this is certainly something that we will be considering. The plan for us that our first in human trial will be conducted in Australia in order for us to take advantage of, first, the relationships we have there with the Lions Eye Institute, and secondly, the tax credit of 43% that Doug mentioned earlier. So we plan on having that first in human study there and transition into our pivotal study where we would include clinical centers in the US, Europe, and some centers through the rest of the world. So yes, those will be in considerations. Right now is our planning phase of where these clinical sites are going to be, what type of our clinical development plan needs to be, and working around developing the fastest path to market from a clinical development plan perspective.
Thank you, May. Next question, what are the biggest risk factors in the RP program succeeding? What worries you?
Yeah, so I think if you look at the benchmarks of why things fail, it's typically, it comes down to efficacy and tox. So will the drug be safe? Now, I'm not really worried because what's really nice is that we've shown that the drug is safe even at dosing levels that are 10-fold higher than what we expect to have to put in terms of efficacy. So it's often defined as like the therapeutic window. How high do you have to get efficacy? And then how high do you start getting tox issues? And in some drugs, that's pretty narrow. And there's this awful trade-off between efficacy and tox. Our therapeutic window is so big that I'm not really worried about it. And so once we go into the animals and monkey, we'll prove that tox threshold is really high. And then the efficacy is the other one and say, all right, how high do you have to boost the drug until you get the result that you want? But like I said, we have so much headroom because the drug is so safe that if we didn't get the efficacy of where we think we're going to get it, increasing the dosing is an easy thing we can do.
Perfect, thank you. Again, we'll wait a few minutes to see if there's any other questions that pull through. I do want to say that, myself, Lindsay here, will be following up with all of you guys individually after this presentation. So if there are any additional questions afterwards, we can always get you in touch with May or Doug as well.
Yeah, absolutely, and would welcome the opportunity to share the story in more depth or hook folks up with some of our colleagues down in Australia. I think, this company has flown under the radar for so many years down in little old Perth, and I'll tell you what, having lived in Australia for the last 7 years, Perth is considered a bit of the backwater, even if you talk about Sydney, Melbourne. So Australia is so remote from the US and Perth's even more remote. So we're just really excited now to share this story on a much bigger stage here in the US.
Thanks, Doug. And actually another question did pull through. How do you know how much drug needs to reach the target to have intended efficiency in RP?
Yeah, so that comes to the testing that we talked in the patient-derived models. So when we get those cells, we put it in, we start to see, all right, what kind of reaction do you get and how high do you think you need? What's beautiful is that patients with retinitis pigmentosa, there are asymptomatic patients. So there are people that have the genetic mutation, but because of some other gene that they have that it's expressing a certain level, they don't get the symptoms. And so we can look at that and say, all right, how far do I have to push the gene expression to get this patient to become asymptomatic? And so we kind of have the answer there. So then it's just a question of how much of our drug is needed to get that level change in the gene expression. So that's why we're testing it in the patient cells. We've done it now on a few patients. And as I mentioned, we're going to be doing more over the next few months. And that'll really sharpen our view of how much dosing we think will be required.
Okay, well, that seems to be all the questions as of now. But again, if anything else comes up, I will get you guys directly in contact with either May or Doug. On behalf of everyone at Family Office Networks, I do want to thank all of our attendees for joining us today. And a big shout out to May and Doug for joining us and presenting.
Thank you.
Yeah, thank you, everyone. And thank you, Lindsay, for organizing.
Thanks, guys. Have a great day, everyone. And we will be in touch soon.